Good afternoon, and welcome to Talks at Google in Cambridge, Massachusetts. Today, it's my great pleasure to introduce Beth Shapiro. Dr. Shapiro is here today to discuss her book, How to Clone a Mammoth, The Science of De-Extinction. We have the science today to restore extinct species to life, but should we? The ethical challenges are enormous. Using DNA collected from remains as a genetic blueprint, scientists aim to engineer extinct traits that evolved by natural selection over thousands of years, into living organisms. But rather than viewing de-extinction as a way to restore one particular species, she argues that the overarching goal should be the revitalization and stabilization of contemporary ecosystems. Dr. Shapiro is an evolutionary biologist who integrates molecular phylogenetics with advanced computational biostatistics to reconstruct the influences on population dynamics in a wide variety of organisms. She is Associate Professor of Ecology and Evolutionary Biology at UC Santa Cruz. Her work has appeared in numerous publications, including Nature and Science, and she was a 2009 MacArthur Foundation Fellow. Please join me in welcoming Beth Shapiro.
Thank you. Well, thank you very much for inviting me to come here and for taking the time out of your busy and hectic days to come and listen to me, especially after David Duchovny, who I'm sure was quite entertaining and probably more entertaining than I can possibly be. So let's just leave it at that, right? I thought I'd begin with a video that kind of explains what I do when I'm in the field. So the quality of the sound is a little bit poor, but the video is kind of neat. So bear with me here for just a sec.
So this is really cool. What we've just found, you can see, is 1, 2, 3, 4 pieces of mammoth bone here. This is part of a vertebra, so you can see how big this is. And the neat thing about this is that these are the small pieces, which means that the stuff is washed downstream. See, these pieces are actually still frozen in the permafrost. You can't get them out at all, which means they're going to be really well preserved. Just heard that big splash of water back there. That means another hole's broken through. Here comes the water. We better get out of here.
So admitting that last part is a little bit ridiculous, but in my defense, the water is really gross. Like, what that is that's active placer mining, gold mining sites up near Dawson City in Canada's Yukon Territory. So during the spring, when the snow melts, the miners collect all the water in these big holding ponds, and then they pump it up to wherever they're trying to wash away that permafrost. And they wash away whatever's thawed and then let it stop for a bit, let the next little few inches thaw, and then wash that away. Their goal is to get rid of all of that permafrost to get to the gold-bearing gravels beneath. But while they're doing that, we find hundreds, thousands, sometimes tens of thousands of these bones that are incredibly well preserved, and we bring them back to the lab and use them for our research. So I'm a biologist, an evolutionary biologist, or a geneticist, or molecular paleontologist, lots of ways to describe that, including some new ones that were used in the introduction just a moment ago. So what does a biologist want to do with frozen bones collected from the permafrost? Well, my research is actually focused on trying to understand how populations and communities and ecosystems respond to rapid, large-scale changes in climate in the present day. So when we see and hear about climate change going on today, often what we hear about are large-scale changes in precipitation patterns that change the distribution of water, maybe differences in storms, and of course, species that are on the verge of extinction. And when we hear about this stuff reported by the popular press, often we're just told that everything is going to hell in a handbasket, right? But if you're a biologist and you want to avoid some of these particularly dire predictions here, you might want to understand or be able to predict in some useful way how the species and communities that we're trying to protect are going to respond for the change in the climate that's predicted for the next decade to century. So if you keep up with the climate change literature at all, you'll no doubt be familiar with this particular graph here, or something similar to this. This is Michael Mann's hockey stick graph. It's called a hockey stick graph because it kind of looks like a hockey stick laying on its side. Here's the end of the stick and the part that goes up here. This is the average temperature around 1960, and this is really just a plot of how that temperature has changed over the last 1,000 years. So you see the temperature remained pretty constant, maybe declining a little bit through time, until a couple hundred years ago when it shot up pretty quickly. Now this plot can be extended into the future, and we see all sorts of predictions of climate increasing, the temperature increasing exponentially throughout the next 50 years to 100 years. Of course, this is not the first time in Earth's recent history that the planet has experienced a really rapid increase in temperature. If we extend this plot backward to the last 50,000 years, we see an interval right here in between around 18,000 years ago and maybe 12 or 13,000 years ago, when the average global temperature increased by about 8 degrees. This here is only about 1.5 degrees. This is an 8-degree increase. And some of the work that we're doing right now is suggesting that this last period of really rapid increase, this is the transition from the peak of the last ice age into the Holocene warm interval that we're experiencing today. This last period possibly happened over less than 100 years, or even as quickly as a few decades. So this is a very rapid period of widespread global warming that's going to be associated with all sorts of changes in temperature and precipitation and changes to the biological species that are living out there. So what I try to do with my research is ask the question, how did species and communities and populations and ecosystems change in response to this past period of rapid climate change? And is there anything that we can use from learning about what happened in the past to make better decisions about how to spend the limited time and energy and resources we have to conserve species today? So the field I work in is called ancient DNA. It's pretty clear what ancient DNA means. It means old DNA, not old people DNA, but actually really old DNA, like mammoths preserved in ice. I work in a place called Beringia. Beringia spans this kind of part of Alaska here, across the Bering Land Bridge into eastern Siberia. All of this white stuff here is just an indication that it's not as deep in the ocean there. So during the ice ages, when much of the water on the planet was taken up into making giant glaciers sitting on top of the continents, this was actually exposed. This was exposed land. And this, of course, is a very important corridor for movement of many species, camels and horses going in this direction, bison and people going in this direction to move between the two continents. So today, Beringia looks like this. I'm actually in that helicopter taking this picture, and I'll show you a picture of that fantastic flying machine in just a moment. Looks like this, but in the past, during the last ice age, it looked more like this, with a very rich and diverse plant community that was grazed on by a rich and diverse herbivore community that was hunted by lots of different types of carnivores. There were mammoths and mastodons at various times, and North American camels, several different species of big cat. There were giant bears, Arctidus, that if it stood up was 16 feet tall, and there was even a giant a giant beaver that was the same size as me, which is the funniest extinct animal. So we fly out here in this fantastic flying machine. You'll notice that there are some windows missing, which is great in case anybody wants to smoke on board in celebration of taking off. I'm not saying that happened, but I'm not saying it didn't. And we stay in 5-star accommodation. As you can see here, this is a picture that I took of my tent by backing up and unfocusing my camera lens so that I could really get a picture of the depth of field of mosquitoes that we were dealing with up there in the Arctic. And we walk along all sorts of places wherever the permafrost is thawing, like these placer mining sites. This is again back in Dawson City. The previous pictures were from the Taimyr Peninsula in north-central Siberia. This is, again, an active mining site where you see them washing down the permafrost. And here are my folks kind of wandering around, waiting to pick up bones that are coming out as the dirt washes away. So in a day out in the field, we'll generally collect anywhere between 5 and 20 bags of bones like this. Most of the bones there are bison. There are also mammoth and caribou and horses, and sometimes we get lucky and get wolves or bears or lions. And we take a piece out of these bones, a tiny piece, that's just a regular Dremel tool with a little cutting disc, and bring that piece back to the lab, grind it up, and extract DNA. And then we correlate the amount of genetic diversity that we observe in any species at one point in time with the size of the population. So in essence, we can use DNA extracted from thousands of these bones to learn how populations were growing or shrinking or moving across the landscape, or whether there were local extinctions followed by replacements of populations coming in from somewhere else. And we've learned an awful lot of stuff over the last couple decades of doing this. For example, we've learned that bison, horses, and mammoths all peaked in population size around 40,000 years ago and then started to decline. And this is interesting because two of the main hypotheses about what caused the megafaunal mass extinction at the end of the Pleistocene, when we did lose mammoths and many other of these big species, were that they didn't like the peak of the last ice age, it was too cold and too terrible for them, or that people turned up and hunted them to death. But if the initial decline begins about 30,000 years ago, that's 10,000 years before the peak of the last ice age, and 15,000 years before any evidence of large numbers of humans in North America. So it looks like we're off the hook for the beginning of these extinctions, although I'm not quite willing to let us off the hook for the ultimate extinction event of many of these species. We've watched carnivore populations grow and move in response to changes in the dynamics of the herbivores that they preyed on, and we're starting to learn why some species, like caribou, survived. They like to live where humans didn't, which is a pretty good trick. And other species, like cave lions, went extinct. And I and colleagues who work on this stuff often get to publish these papers in pretty high-profile journals. They get lots of press attention, and people care. People are really interested. They call. I'm often really excited to talk about what we've learned and how it applies to present-day problems in biodiversity conservation. But they only ever really ask me one question. And it's kind of annoying. And since there really is no soundbite-sized response to this, I decided to write this book, right? So the field of bringing extinct species or extinct traits back to life has come to be known as de-extinction. Now, we are all partly familiar with de-extinction because we were there when it happened the first time, and we know what happened. We know it all went off without a hitch. Everything was perfectly good, right? Yeah? We remember this? But today we're not going to talk about dinosaurs because we can't clone dinosaurs. There are no dinosaur bones that contain DNA. They're all rocks. There's no DNA in rocks. Regardless of what you read in high-profile, nice publications like Science and Nature, there's no DNA in dinosaurs. You heard it here, right? So how are we going to bring back a mammoth? We know there's DNA in mammoths, right? The first step, the first thing that everybody thinks we're going to do, the first process that's proposed is let's clone it. Let's clone a mammoth. Well, cloning is actually a specific scientific process, a specific type of work that's actually known as somatic cell nuclear transfer. So in our cells, we basically have two different kinds of cells: germline cells, which are sperm and eggs, and somatic cells, which are everything else. What normally happens, a sperm and an egg come together, fertilize, create a small thing, a zygote, and that differentiates, and that particular cell that they create has the power to become any type of cell in a whole body: hair cells, skin cells, heart cells, liver cells, every type of cell there is. Somatic cells already know what their job is, and they can't do anything else. So the idea of somatic cell nuclear transfer is to trick a somatic cell into forgetting all the instructions necessary to be that type of cell, and to revert into a state where it can actually then become every type of cell in a body and create a whole being. So the most famous and the first example of somatic cell nuclear transfer was an experiment in the mid-1990s that brought us Dolly the sheep. Probably everybody's heard of this, right? This was work by the Roslin Institute in Scotland. And they took a somatic cell, in this case it was a mammary cell from an adult sheep, a ewe, and they took those cells and basically starved them. They starved them of nutrients in a dish in a lab. At the same time, they took an egg cell from a different breed of sheep, and they removed the nucleus, all the genetic material from that unfertilized egg. So you had an egg with nothing in it and a bill starved cells right here. Put them next to each other, zap them with electricity, the membrane bursts, the nucleus here dumps into this cell, zap it again, and the proteins in that egg somehow cause the genetic material from that somatic cell that's a bit stressed out to forget all the instructions and turn into the type of cell that can become everything in a particular animal. This is then implanted in a surrogate host who is yet another species of sheep or breed of sheep, and eventually Dolly was born. And Dolly was a genetic clone of the mammary cell donor and not of either the egg cell donor or the surrogate mom. Easy. So how do we do it with a mammoth, right? Well, we go out in the field and we find one of these fantastically preserved mammoths, and we remove some cells, and then we stress them out, and then we stick those cells in the nucleus of an egg, and it does that little magic-y thing, and then we stick it inside an elephant, and it grows up into be a baby, release it, and it's all good. Right. Right. Here we have our first problem. So we do find a lot of incredibly well-preserved remains up in the Arctic. This is an example. This is a horse jaw that we found up in the permafrost near Dawson City that dates to sometime in the last 100,000 years or so. This is one of the very many well-preserved mummies that's been found in Siberia. And a couple summers ago, there was a report of this incredibly well-preserved mummy that was associated with what looked like a viscous red material that they thought might be blood, right? But regardless of how well-preserved these things are, there is not going to be a living cell to be found in any remains of any mammoth. Once an organism dies, the cells and the DNA within them begins to get broken down into smaller and smaller pieces almost immediately. First, it's enzymes within our own body that chop up the DNA. If you're a mummy, you might have the guts burst, and then all of that microbiome that lives in there is going to circulate throughout the system. And those are living microbes that will just catabolize, eat up that DNA. Sun is particularly bad. UV light will hit those mummies just like it hits us when we're alive and we walk outside. But unlike us, the mummies are dead or the bones are dead, so there's no proofreading mechanism to fix the damage caused by solar radiation. Water, hydrolysis, oxygen, all these things are terrible for DNA survival. And in fact, the DNA begins to break down immediately and is eventually broken down to a state where there's nothing that's recoverable. Where there are no living cells, there's no living genomes, there's no intact DNA, we cannot clone a mammoth. Thank you for coming. I'm just kidding. Obviously, I'm just kidding. So what's the next step? So we all probably heard last week, as there was a lot of press attention, that a group, an international group of scientists, but mostly a group that was in Stockholm, led by Lova Dahlen, had published the genome sequences, complete genome sequences of two mammoths. One mammoth from Wrangel Island that was around 3,500 years old, and another mammoth from Siberia that was somewhere in the realm of 40,000 years old. So now what we have is this nice big list of A's, C's, G's, and T's, the nucleotides that make up DNA, that will spell out the instructions to make the genes that make the proteins to make a mammoth, to make it how it looks and how it behaves. So surely the best thing that we can do to bring a mammoth back to life would just be to synthesize these long stretches of DNA and then get them into the chromosome and then put the chromosome into the egg cell and then, do the thing with the going around and making the mammoth, right? Yeah, that. So the problem with this is that a complete genome, well, there's lots of problems here, but let's just start with this. They said it was a complete genome, but in fact, it's not really a complete genome. In fact, we don't actually know the complete genome sequence of any vertebrate animal, including humans. And I know that that sounds a little bit strange, since we've been saying for quite a long time that we know the human genome sequence. And to be fair, we do know most of the human genome sequence. We know at least all the parts that contain genes, the important parts that make the proteins that make us look and act the way we do. But there are parts of the human genome and of every other mammalian genome and vertebrate genome that are extremely hard to sequence through. These complex patterns of highly condensed repeat things, often near the center of the chromosomes and near the end of the chromosomes, that there isn't any existing sequencing technology that can get through. And this means that we don't know the whole sequence. We don't know how long these are. We don't know if they're important. We don't know whether there are any genes in them. We don't really understand quite yet what they do. But the truth is, we don't have the complete sequence. For mammoths, it's even harder than that. And there are two reasons why it's harder for anything that's extinct, in particular we're talking about mammoths here, than it would be to assemble a complete human genome. The first is that the sequences that we recover from these bones are very short and damaged, and this is because of all of the stuff that I just talked about: solar radiation, enzymes from in the body, and from microbes that are in the soil that will get into the particular bones after they're buried and eat up the DNA. All of this stuff is acting to break those DNA sequences down. So if you imagine that I could get modern DNA out of something that looks like a nice big party streamer, ancient DNA looks more like confetti, right? But not confetti that's in that good of a shape, right? More like confetti that you might find on the ground in the gutter the day after the parade, right? When it rained the night before. And maybe there were elephants in the parade and flocks of passenger pigeons flying overhead. You know, not good. It's not good. The other problem is that the samples are full of all kinds of stuff in addition to mammoth DNA. So if you imagine that I took a piece of my own hair or a cheek swab and extracted all the DNA in there and then sequenced everything I got when I extracted DNA, nearly everything that I could get out of there would be my own DNA. That's because I'm living and my DNA is in pretty good condition and there's not that much that might be contaminating my DNA sample. In 2006, I was involved with the group that used the new sequencing technology, next generation sequencing, to shotgun sequence, that's what we call sequencing everything we get out of a DNA extract, a mammoth bone for the first time. And this is what we got. About half of the sequences that we recovered from that sequence mapped to mammoth DNA. The rest of it was stuff like soil bacteria, environmental microbes, other stuff that was probably also soil microbes that we just hadn't sequenced yet, so they weren't in the database for us to compare the data to. Some contamination. We often find human, dog, domestic cats, and bird sequences in our extracts. That's not that surprising. So about half of what we got was mammoth. We were pretty bummed about this, right? We thought, whoa, half, that's terrible. Like, how are we ever going to be able to piece together a complete genome with only half of what we're getting is actually ancient DNA? It turns out, several decades, well, a decade later, we know that this was actually an exceptionally well-preserved sample, and that most of the remains that we recover have less than 10% of the DNA that we want is actually the DNA from the host. If you've heard of the Neanderthal genome that was published recently, the first Neanderthal genome sequence, they used a couple of different bones, and none of those bones had more than 1% Neanderthal DNA in them. That's pretty standard, especially when you're not coming from a kind of cold region. So what we have is this kind of junked up bit of confetti, if you can imagine confetti that looks more gross than this. And what we really want are just the purple bits, right? So how do we go about finding the stuff that we want from amid the entire mess of stuff that we actually get? How do we tell what DNA is mammoth when most of what we're getting is actually just soil microbes that we really want to throw away? What we do is we map the data that we have, these broken little tiny pieces of fragments, to the already published complete genome sequences of living animals, or complete-ish, right? They're in good shape. They're much better than anything that we would be able to generate used from an ancient sample. So for the mammoth, we would map it to an Asian elephant, and for the Neanderthal, we're able to map it to a human. We take these broken fragments and we match them where they go, using this genome sequence as a guide to figure out where they go. One of the key problems that arises immediately when you think about this is, what if something weird has happened in the evolutionary time between when a mammoth lived and when the Asian elephant lived, since that common ancestor? If something weird happened, like a big duplication, there's a whole new bit of DNA, or something changed a lot, right? That would be missing. We wouldn't have that part of the genome on our map. We would think that whatever it was of mammoth DNA that matched that real part of the mammoth genome, because it's not part of the elephant genome that we're using as a scaffold, was probably just environmental DNA, and we would throw it out. So it's harder to generate a complete sequence for something that isn't alive, where we can't generate the genome sequence by itself. And one would think that if you're trying to identify the parts of the genome that are most important to make a mammoth look more and act more like a mammoth instead of like an elephant, those might be some of the important places to look. The other problem is that having the DNA sequence is not the same thing as having a living cell. We're getting better at being able to synthesize these long strands of DNA that we might use to generate a complete sequence, but we don't know how to wrap those things up properly and get them into a chromosome in a state where they're actually going to make genes and make proteins. And we don't know how to get chromosomes into a cell in any logical way. So it looks like if this is the pathway, plan B is what we're going to use to bring a mammoth back to life, we're stuck at that first step, and that's not going to work either. Of course, there's another way. So the last plan, and this is the one that really is at the core of what people are thinking about for de-extinction, is to engineer a mammoth. And this is really similar to just thinking about cutting and pasting your DNA. So you find a part of the genome that you want to swap out, say some gene that's different between elephants and mammoths. You cut it out and paste in the version that you want. So mammoths and Asian elephants diverged from each other something like 6 million years ago. We now have genome sequences from both of these sequences, and we can start combing through those genome sequences to look for places where the two species are different from each other. Sequence a whole bunch of Asian elephants, finds a place where they're all the same as each other, a whole bunch of mammoths, and see where they're all the same. And if these two are different, then that's probably kind of critical to making a mammoth look and act like a mammoth. So imagine then you had a little machine that you could use, a machine that you could send into the genome of an elephant cell and identify exactly that place that you want to change. And also you could give that little machine a package to take with it, which was an artificially synthesized bit of mammoth DNA that it would take with us to this place in the genome. And that machine could then cut out the elephant version of the gene and swap the mammoth version gene in there in its place. Well, we have that machine. That machine is called CRISPR-Cas9, and this is something that you might have heard a lot about in the news in the last couple of weeks. There was a Chinese team recently that used this particular enzyme. This is a naturally occurring enzyme, not a machine that we've built. It's something that bacteria and archaea use to protect themselves from invading diseases. People have figured out how to use this, to harness this machine, to identify parts of the genome and cut it out. And this Chinese team had done that with human embryos, raising all sorts of ethical issues about this incredibly powerful new technology. So this machine here, this molecule here is your little machine. This is an enzyme. And here's the part of it that recognizes a specific part of the elephant genome sequence. We send in there a bit of synthesized mammoth DNA. The machine here chops it in half. It's like a molecular scissors. That's its job. Of course, DNA in a cell doesn't like to be in half, so it has a couple of different mechanisms that it uses to fix those double-stranded breaks. We harness the cell's own repair mechanism and stick in there that synthetic piece of mammoth DNA. So we end up with an elephant that's just a little bit mammoth-like. Right. So what do we change? I said there are about 6 million years of evolution separating these two species, and indeed there have been some efforts so far, within the last couple of weeks even, to catalog the differences that existed between these two guys. One of the first differences that was found was a work that was done by Kevin Campbell's lab in Manitoba in 2010. They found that there were only 3 differences, 3 changes that needed to be made to change the Asian elephant red blood cell, the hemoglobin component of the red blood cell, which is responsible for carrying oxygen around the body, into the woolly mammoth version of the red blood cell. And they made these changes in cells in the lab, and they were able to then do a little tissue culture experiment, a gene expression experiment, to try to figure out what those differences did for that version of the hemoglobin protein. And they found that when these changes were made, the hemoglobin was much better at carrying oxygen around the body at low temperatures. This is clearly something that is an adaptation that arose in mammoths as the tropically adapted ancestor of these two species moved out of the tropics and into the Arctic, right? So this might be a powerful new first approach to going about trying to create or change an elephant genome so that you could create a mammoth. So what else should we change? Well, there's a team that's nearby here that's led by George Church that have been working on this for some time. And using this CRISPR type of genome editing, they've so far been able to swap out 14 different genes. Every one of them that they've tried so far has worked with this CRISPR technology. These genes are associated with increasing thermal regulation, maybe thicker subcutaneous fat layers and thicker hair, or the red blood cells. Also things that are associated with diet and metabolism, all sorts of things that one might think were important to making an elephant look and act more like a mammoth. And so far, they have succeeded in creating something that is about a small fraction mammoth in a dish, in a lab, which of course is a very long way from creating a living, breathing mammoth. But it is hopeful, and it does say that with this genome sequence, which we do have for the most part, and with this CRISPR-Cas engineering technology, which we also have, we can then create an elephant cell that's alive, that contains a few mutations that make it look and act more like a mammoth. And at that point, we can go ahead and use cloning, the traditional technology used to bring something back. Of course, the next step here, creating a surrogate mom, growing it up, putting it in the environment, I like to call it phase 2 of de-extinction, is probably even harder than what we've been able to accomplish so far. And when you read about de-extinction or hear about it on the radio, often there's a focus on this first part, phase 1, with the thought that just because we can get this far means that we are super close to being able to bring a mammoth back to life. But that is really, really not the case. We are far from being able to create a mammoth. So the next thing we would have to do in this step would be to find a surrogate mom. So for many species, this might be easy. If they have very close evolutionary relatives that are still alive, then it might be relatively straightforward to finding a surrogate mom that could carry an edited embryo or an edited embryo to term. But the more evolutionary distance accumulates between the living species and the extinct species you're trying to bring back, the harder this is likely to be. There may even, in some cases, be some unpleasant constraints associated with trying to do this. I know I say here that you probably don't want a photo, but you're a bunch of computer scientists, so you probably do want a photo. So here is one. You're hoping for something grosser, right? Yeah. I knew that. I'll try to find something grosser for next time. This is a stellar sea cow. And this stellar sea cow, this is a foot. There's a scale bar right there. This is a big animal. Stellar sea cow went extinct a couple hundred years ago. We hunted them to death. One of these guys could feed a crew of 30 or so men for a couple of weeks. So you can see why potentially they were nice prey for people who were out on the ocean. They lived along the coast of California, up across the Aleutian Islands and onto the Commander Islands. And they went extinct really recently, a couple hundred years ago. The closest living relative to the stellar sea cow is a dugong here, right? And if one were to estimate the size of a newborn stellar sea cow by looking at the ratio of newborn manatee and dugong to the size of their mom, a newborn stellar sea cow would be larger than its mom, which just wouldn't be pretty. So in some cases, there may be some physical constraints, technical problems, technical challenges associated with this part of phase 2. It's also pretty clear by now, and becoming more and more clear through time, that we as organisms, and every organism, are more than the sum of just our ACs, Gs, and Ts that make up our DNA sequence. In fact, we are a product of both our genome sequences and the environment in which we live, and that environment begins in the womb. The same would necessarily be true for an elephant or an edited elephant that was attempting to be a mammoth. Asian elephant and mammoth genomes are already, without thinking about anything, about 99% the same, about the same as we are and with chimpanzees, right? And so if you put something that's tiny, tiny bit changed inside a mom who has pretty much the same genome and pretty much the same genes, and the developmental process here is going to be controlled and affected by her hormonal environment and her diet and what's going on, how do we know that the changes that we've made are going to have any effect whatsoever on what this animal looks like? And once it's born, it's going to be born to and live in an elephant society. Elephants eat the dung of their moms just after they're born in order to establish the microbial community that they need to break down the food that they eat. This newborn mammoth-elephant hybrid would eat the dung of an elephant so that it could establish an elephant microbial community. We know how important the gut microbes are in determining how we look and how we act and how we feel. Why would the same not be true for an animal like this? It would then join an elephant society and eat whatever food elephants are fed in captive environments. And you know, all of these things are going to affect the way that this animal behaves and the way that it looks. This is something that's unquestionable and yet something that we haven't really questioned. And speaking of captive environments, this in my mind is probably the biggest challenge associated with mammoth de-extinction. We know that elephants don't fare particularly well in captivity. They often fail to reproduce in captivity. If they do, they injure and sometimes even kill their offspring. Until we have figured out how we can meet the physical and emotional, psychological needs of mammoths in captivity, I think that we should not be using mammoths for this type of research or keeping them in captivity. Maybe for other species, these particular types of concerns are less. Birds, we may not need some bird species might not have very much parental guidance, so maybe it's ethically simpler to bring birds back, but there are, of course, technical challenges of bringing birds back that we don't have when we're dealing with mammals. So, you know, there are many different ethical considerations to be aware of, but again, this is something that I think not enough attention has been paid to. And finally, it would be remiss of me not to point out or to acknowledge that the world has changed a lot in the time since many of these species have gone extinct. And bringing something back and introducing it into an environment where it doesn't live is definitely going to affect those environments. Environments are not stable things. A species doesn't go extinct, leaving this giant gaping hole into which we could just stick it back. They'll change, they'll adapt, new species move in, everything changes. This is the range of the passenger pigeon, which is one of the species that's been proposed to bring back to life, right? Where would a passenger pigeon, a bird that flocked in the billions and fed on forests, killing huge swathes of forests all at once, where would it go if we were to bring it back? Clearly, the ecological implications of de-extinction need to be considered carefully for any species that we're considering bringing back before we go down the road to actually making it happen. So de-extinction, can we do it? Not yet. We can't yet. But the technology is increasing rapidly, both in phase 1 and in phase 2, and there will come a time where we can do this for some species. So maybe we should start focusing more on the question, should we? And as somebody who's written a book about this, it's clear that I should have a very informed and direct opinion about this, and I do. I don't know. I don't know. I honestly don't know. There are a lot of different species that have been proposed as candidates for de-extinction. This is the heath hen that used to live in Martha's Vineyard, mammoth, passenger pigeon, thylacine, stellar sea cow, the gastric brooding frog, the bucardo, and a type of ibex that used to live in the Pyrenees. All of these things have been posed by groups who are interested in working them to be resurrected. But I think that the most important thing to do in deciding whether we should proceed with any of these species or any other species that someone might want to bring back is to really try to understand why. Why might we want to bring a species back that's been extinct for a long time? Why is this such a compelling area of research? Why are so many people so fascinated by this idea? And to answer this question just in the last few minutes here, I'm going to return to mammoths, and I'm going to kind of ignore the fact for a moment that it's not possible to do, and that ethically it's probably a terrible idea to deal with elephants and to bring mammoths back to life, and talk about why we might want them back. Because I do think there are a couple of compelling reasons to think about bringing mammoths back, or at least creating an elephant that looks and acts a little bit like a mammoth. The first is ecological. So there's a place up in northeastern Siberia called Pleistocene Park. It's been established in about the mid-1990s by a scientist with the Russian Academy of Science named Sergei Zimov. He started buying up land near his home at the Northeast Science Station and preparing a park for the resurrection of Ice Age animals. So far, he has bison, imported from Canada, some species of horse, 5 different species of deer. And he's been doing some experiments where he's keeping them out of some parts of Pleistocene Park and letting them graze on other parts of Pleistocene Park to see what the effect of having those animals there is on the ecosystem, the environment itself as a whole. So here's a picture that Sergei took. This is very early spring. This is the side that doesn't have any grazing animals, and this is the side that does have grazing animals. And what you see here is a really stark contrast between the type of vegetation that's there. Here we have pretty much only one species of grass. It's very low productivity, really can't support grazing herbivores in any way. And over here, remember this is early spring, so the grass hasn't had time to regenerate. You still see lots of green, and you can't really tell, but there are a bunch of different species of grass that are there. What he's found is that these animals, by their presence alone, moving around, turning over the soil, recycling nutrients, distributing seeds, have essentially created their own habitat. They've made the habitat that's necessary for them to survive. In fact, they've made so much that even after the very cold Arctic winter, there's still some green grass there left for that community to come back the next year. And he's observed not only that these animals are happy and healthy and living in these areas, but that other species have come to visit the park because it's such a rich source of food, things like saiga antelope, an endangered species. Essentially, these animals, with their presence, have recreated that rich tundra grassland that used to be there, potentially making habitat and saving species that are in danger of extinction today because this is all they've got. The next reason is more emotional. I think few of us are willing to accept the idea of a world that doesn't have elephants. But elephants are an endangered species. Asian elephants are in trouble. Their habitat is declining. We're having a difficult time stopping poaching. Human populations are growing, and every year fewer and fewer Asian elephants exist. What if we could use this technology not to bring mammoths back to life, but to create elephants that are capable of living in a more diverse environment? Elephants that could live in temperate zones or Arctic zones, moving into the types of habitats that were once occupied by mammoths. We could use this technology not to bring something that's gone and has been gone for a long time back, but really to save a species that's still alive today, but in danger of extinction. Let's not use this crazy technology to de-extinct mammoths. Let's use it to save elephants. And that's what I think the most compelling use of the technology that's being developed here really is. Not to bring stuff back that's gone, but to change and manipulate and give a genetic booster shot, if you will, to populations and species and communities that are in danger of going extinct because of our actions today, because of climate change, because of decreases in habitat that evolution itself alone can't keep up with. This, in my mind, is the most powerful use of de-extinction technology. Thank you for your attention. I'm happy to take any questions, if anyone has any.
So my first question comes back to what you said at the very beginning, which is this is not what your field was. Has the question caused you to become interested in this question and change what you're working on, or is it just caused you to write this book that you're now tour about?
Yeah, no, I'm definitely more interested in it now. And actually writing this book was an experience in becoming more and more interested. We now have a couple of different projects where we're hoping to use ancient DNA technology to learn what the diversity was in populations that were extinct in the past, that not extinct, but where recent changes in population size, like black-footed ferrets in the Central Plains, for example, are nearly genetically depauperate. They're all almost the same, and that's because of a recent really tight population bottleneck and near extinction. These guys are now in trouble. There's a disease that's killing them off. It's not something that we can do yet, but it would be possible to look at black-footed ferrets that are preserved in museum collections or living cells that they have in the collection at the Frozen Zoo in San Diego and learn how much diversity there was in the past, and then use this technology to inject that diversity into living populations of black-footed ferrets. I'm also really interested in birds. There are tons of species of birds that are in danger of going extinct, and yet the biggest technical hurdle here is that we can't clone birds. There's no way to actually get that egg cell at the point where it's ready to be fertilized, but it hasn't yet been fertilized because of the peculiarities of the bird reproductive tract. And so there is no way to clone birds. There's a different type of technology that one would use to change the genome sequence in birds. And we've been working with other groups to try to refine that technology for endangered bird species. It's kind of neat. When a bird chicken egg is laid, for example, the germ cells, or the primordial germ cells that are going to go on to become sperm and eggs, aren't actually in the gonads yet, because the gonads don't exist yet, but they're migrating around the outside of that embryo to the place where the gonads will be. At that point, you can stick a needle in the egg cell and suck out some of those germ cells, primordial germ cells, without harming the embryo or the egg. And you can put those into a dish, and you can actually edit those primordial germ cells. And then you can stick them back into another egg as it's just been laid, and they will migrate around the embryo to become those primordial germ cells. So the egg, when it hatches, will be totally normal, but its offspring will have the edits that you've made. This is a really cool and potentially powerful technology that's really at the early stages of being developed.
Very cool. So you pose the question as, can we and should we? Do you not feel like we live in a world where it is inevitable that probably within the lifetime of people in this room, people will actually decide to do this, and there will be baby elephants with a little bit of woolly mammoth, or baby humans with a little bit of Neanderthal, or whatever your favorite choice is, as one of these inevitable...
Those are two particularly hard choices, right? There are a lot of technical hurdles with both of these, and with Neanderthals, there's a big ethical question there. Neanderthals are people too, right? So how are you going to get informed consent to bring one of those guys back to life? That's a pretty touchy thing. I don't think we should go there. Whether we do or not, I don't know, scary, but there are a lot of scary things that can be done with CRISPRs, so I don't think we should talk about those either. Mammoths, some of the biggest hang-ups are actually in the cloning process itself. Every time an elephant gives birth, the hymen actually grows back, and the hymen has a tiny little hole in it that's just big enough for the sperm to get through, and it sits there during the pregnancy to actually maintain the pregnancy. It's a big, heavy thing, right? And it's a 2-year pregnancy, so gotta hold it in there for 2 years. There certainly is no way to insert a developing embryo into the uterus of an elephant without destroying that hymen. I think that this stuff is going to involve, growing elephant eggs on the backs of mice. I know that there are groups that are actually working on doing that, and maybe completely artificial wombs where the entire gestation process takes place outside of a species. And all of that stuff might be developed, probably not for de-extinction, probably because we want to use it for humans. There certainly isn't any money being invested into developing technologies for the purposes of de-extinction. But yeah, do I think that somebody's going to try? Yeah. More likely, somebody is going to achieve what George Church's lab achieved, and then the press is going to say, George Church cloned a mammoth, which is not what George Church said at all, right? And to be fair, there was only one media outlet that said that, so, and I won't say what it is since I know this is going on YouTube.
Okay, so to play devil's advocate here, so let's say that we succeed in genetically engineering elephants so that they can survive in different habitats than they naturally live in now. That feels to me like, going up to the elephant and saying, well, we've destroyed your natural habitat here, but we found this other place in North Dakota where you can live. And so we're going to genetically re-engineer your offspring and have them go do that.
I'm not saying that this should be a substitute to trying to protect the species that are alive and endangered today. But I think that we have come to a point where we're playing conservation triage, and we might need to reach deeper into our arsenals and find the most powerful weapons we have to stop these species from going extinct. I would rather have elephants in Africa, but I would rather have elephants in North Dakota than no elephants.
So, but on the one hand, I don't like to anthropomorphize the argument, but on the other hand, if we did anything like that with humans, it would be ethically abhorrent on a number of fronts.
If we tried to save humans from going extinct, it would be ethically abhorrent?
Or if we relocated them and said, well, we're genetically engineering you, so that's going to be okay, without consent from the participants.
You're making an argument that says we should focus on saving elephants where they are, and I totally agree with you on that. I completely agree that we should do that. But I'm saying that that's not enough, and it's probably not going to be enough. There are situations we can't control. Look at the rhino, right? We haven't been able to save the rhino, right? The habitat where it lives is something that we can't deal with because of current political situations. What if we relocate elephants to North Dakota, and then in that time, when they're gone, completely fix the political climate and the expansion of human populations in Africa, and then we can put them back in Africa? Would having them live in North Dakota for a short period of time be a terrible thing to do if our ultimate goal was to create a native habitat for them to thrive as a living species rather than something that's just gone?
Sure.
How do we actually know what modifying which bits is going to help elephants live in North Dakota?
We have no idea, and this is really part of the process. So we now have these genome sequences, right, which is like somebody handing you 2 massive phone books for New York City and San Francisco, but without any names and addresses in them, right? The next step is trying to figure out where the genes are, what the genes do, doing these experiments like George Church and his lab are doing, where they switch a little bit out and then they test those cells. You can actually perform some assay in tissue culture, in culture, to figure out whether those changes have done something and what those changes might be before using them to go on and make an animal. But yeah, we're very much at the early stages and trial and error. We don't have to make animals, though, that are somehow unfit. We can do those early stages in the lab.
I guess I was thinking, like, even later, like, if we know how to modify the blood cells to live in a colder climate, how do we know how much of that we need to do, or whether that's going to be helpful, and whether the elephants are even going to survive in North Dakota?
Yeah, we don't know, do we? But yeah, we'll know more after we've tried than we knew before we tried. All right, thank you very much for your attention.